Hello, good afternoon, friends. Myself, Richa. So, today I'm going to talk about lac mutants supports the operon model. This question is quite confusing in the exams. So, basically, two things which you have to remember. So, first of all, what is mutation? So basically mutation in the genes, it alters the product of the gene, or it can prevent the gene from functioning properly or completely. Okay, so here the lac operon, this is lac operon, and its regulators were first characterized by studying mutants of E. coli, Escherichia coli, that exhibited various abnormalities in lactose metabolism. So some mutants like this I negative, or you can say OC, these are mutants. So if the mutation happens in the lac I gene, here you can see here I negative, this is I negative, means the repressor is not getting formed properly. See here, you can see the repressor here. So the operator binding region of repressor is altered here. So no binding occurs, so transcription will proceed further. So if there is some problem in the repressor, right? So what will happen here? There is either the production of a repressor polypeptide is altered, or it produces a polypeptide that cannot bind to the operator sequence here. So it cannot able to bind. That's why the transcription will happen constitutively. Means it will go proceed further and further and further. It will not stop. Second mutation occurs in the operator one, that is called as OC here. Here what happens, the repressor is okay, like here it is I positive here. So repressor, whatever it is formed, it is okay, but there is a problem in the operator one. So nucleotide sequence of operator gene is altered here, so no binding occurs. So if the repressor is okay also, but it cannot be able to bind to the operator, so once again the constitutive expression occurs, means transcription will proceed on and on and on. Okay, so there are two things, constitutive mutants, you can say, both are like this, right? So both of these constitutive mutants results in the transcription of the gene. So here, and there is one more mutant which is called as IS, which is not described here in the diagram. IS, that is called as super suppressor, right? So in that what will happen in the case of super suppressor, it prevents the repressor polypeptide from binding to the lactose. So thus it will bind to the operator and be non-inducible forever. So it will be constitutively repressing. So here the no constitutively repressing the lac operon. So no transcription will happen here. So two things you have to remember where the constitutive expression is happening. If there is I negative, otherwise OC case also. If there is mutation in the operator gene, then also constitutive mutation will occur. If there is mutation in the I negative gene, then also constitutive expression will occur. And if there is IS, that is here, that is super suppressor. So in that case, in this mutant, it constitutively repress the lac operon. Whether the lactose is present or not, it is not, means it doesn't depend on that. Okay. So these are the things which you have to remember. Thank you.